Okay, welcome to part 3 of my chapter 5 lecture series. We left off at osmosis. Osmosis is the diffusion of water across a selectively permeable membrane. So since it's diffusion, it's also a form of passive transport. You need to know some vocabulary here. Solute is just a substance that's dissolved in a liquid solvent. A solvent is the liquid that dissolves. A solute, water, is the universal solvent. A mixture of a solvent and solute or solutes is called a solution. So osmosis is a little bit tricky because when we get used to talking about diffusion, we're talking about the movement of the solute particles or molecules usually. Well, in osmosis, we need to kind of turn it around in our head and think of the movement of water. So in this system, and here are some other vocabulary words, hypotonic, hypertonic, and then we'll get to isotonic. So in this system, you have this little U-shaped container with a selectively permeable membrane that prevents the movement of the green sugar molecules. It prevents the movement of those across that membrane. And so in order for this system to achieve equilibrium, something needs to move, but since the solute can't move, it's going to be the water. And so on the left-hand side, you have a hypotonic solution, meaning that the concentration of solutes is lower compared to the solution on the right, which is hypertonic, meaning that the concentration of solute is higher compared to this one. And so what's going to happen is that osmosis is going to occur, and the water is going to move from the left to the right. So in order to balance out the solute concentration, creating isotonic solutions. Isotonic is when two solutions have equal solute concentrations. And so the way that this gets confusing is that we're looking at which way water is going to move. It's still passive transport, so the thing that's moving is still going to be moving down its concentration gradient. And so over here, where you have more water relative to solute, the concentration of water is higher compared to over here, where you have more solute molecules compared to water. So in order to reach equilibrium, osmosis has to happen in this direction. Okay, so here are those terms again: hypertonic, hypotonic, and isotonic. They're relative terms and can only be used when you're comparing two different solutions. A hypertonic solution has a higher solute concentration compared to another solution. A hypotonic solution has a lower solute concentration compared to another solution. And if you have isotonic solutions, then you have two solutions that have equal solute concentrations. Okay. In order to survive, cells have to have a way to regulate water that is lost and taken in. Animal cells need to have a way to be able to survive in hypotonic or hypertonic environments. And in that process is called osmoregulation. It just means the control of water balance. Osmoregulation. And this is a figure from your book, and it shows what happens on the top row to animal cells placed in different solutions compared to plant cells placed in different solutions. So if you have an animal cell, and this is representing a red blood cell, placed in an isotonic solution, then the net in and outflow of water is equal. You have no net in and outflow. And so that is a healthy, normal animal cell. If you place an animal cell into a hypotonic solution, it's going to take on water to try to balance, to try to reach equilibrium, and it will lyse or rupture because it just gets filled up so much and there's nothing to protect the cell membrane from rupturing. And then if you put an animal cell into a hypertonic solution, in order to try to achieve equilibrium, water is going to leave the cell and the cell will shrivel. In contrast, plant cells, as we learned in earlier chapters, have cell walls. And if you put a plant cell into an isotonic solution, it will be flaccid, which means slightly wilted. If you put a plant cell into a hypotonic solution, it will be able to take on water, and then it will exert the plasma membrane that's full of water will exert pressure against the cell wall that prevents it from rupturing or lysing, and that situation is known as turgid. That's normal for a plant cell. That's when, when you forget to water your plants and they get all wilted or flaccid, that's abnormal. When they have plenty of water, they're turgid or normal. That's what helps them to stand up and look pretty. So that's in a hypotonic solution. This is normal. If you place a plant cell into a hypertonic solution, just like in the animal cell, water is going to leave the cell in order to try to achieve equilibrium, and the plasma membrane will shrink away from the cell wall and can lead to a fatal condition called plasmolysis. So in plants, like I said before, they have rigid cell walls that prevent them from lysing or rupturing in hypotonic environments. Plant cells are the most healthy in a hypotonic environment where you have a net inflow of water. Okay, so this is a really good checkpoint. And if you answer all of the checkpoints in this lecture, I will give you 10 points on your test. But you have to answer all of the ones in all 3 parts of my Chapter 5 lecture. An animal cell shrivels when it is blank compared to its environment. And that's looking for hypo, hyper, or isotonic. Checkpoint 2 is the cells of a wilted plant are blank compared with their environment. It's looking for hyper, hypo, or isotonic. Okay, this is just a picture. A wilted plant is flaccid. A healthy, upright plant is turgid. Okay, so moving on to our very last form of transport, active transport. Active transport, in contrast to passive transport, just means that it requires cellular energy to move a substance in or out of the cell. Cellular energy, as we discussed before, is ATP. And the reason that the forms of active transport require ATP is because in this type of transport, solutes are moved, or substances are moved against their concentration gradient, which is the opposite of what we talked about in passive transport. Active transport is important because sometimes cells need to maintain their internal environment in a way that the solute concentration of certain solutes are either higher or lower, so either hypertonic or hypotonic compared to what's outside or compared to what's in the environment. Okay, so this is just a picture from the book showing, using ATP to move a solute molecule into the cell, even though there's already a higher solute concentration, okay, inside the cell. And here it's been moved in, and so that's active transport. It's moving it against the concentration gradient from an area of lower concentration to an area of higher concentration. Exocytosis and endocytosis are just 2 major categories of active transport. Exocytosis means moving something outside of the cell, and endocytosis means moving something inside of the cell. In both cases, the mode of transport is using a membranous vesicle. Know the word vesicle. A vesicle is just like a little packet or sac that is surrounded by the same material that a cell membrane is made out of. It's a phospholipid bilayer. And so in exocytosis, a vesicle encloses whatever material the cell needs to get rid of, and that vesicle fuses with the plasma membrane and exports materials. In endocytosis, the cell membrane will actually move in such a way that it takes in substances from outside the cell and brings them inside the cell. There's phagocytosis, which is also known as cellular eating, and that's when cells take in particles, large particles. And then there's penocytosis, where they take in liquid droplets. This is a picture showing how vesicles are used to transport materials. This is a vesicle surrounded by a phospholipid bilayer. It just encompasses the material to be exported. It fuses with the cell membrane, and then the substance is released outside of the cell. In endocytosis, it's very similar. It's just moving substances in the opposite direction, and you end up with a vesicle inside the cell. That is it for my Chapter 5 lecture, and I will be uploading lectures for Chapter 6 and Chapter 7. Thank you very much for listening, and remember, if you answer all of the checkpoint questions and send them to me in an email, I will give you 10 bonus points on your upcoming test.